Hello. So again, good afternoon. Um, I just want to show you something. Can you see? Can you see it?
Yes po.
Yes po.
Can you tell me what culture media is this one? Anyone?
Chocolate po.
Chocolate. Mm, or who else can make a guess? Kung ano to? Take a look. Ay, hindi ko pinibuksan. Ano kayang culture media to? Any guess? So, this is a McConkey agar. Ito yung sinasabi ko sa inyo, McConkey agar. So, McConkey agar is an example of selective and differential, and makikita nyo talaga that it is color pink kasi nga the organisms is lactose fermented. The organisms are lactose fermenters, and ito yung examples ito yung example ng na Klebsiella pneumoniae. Yan. So meron pala tayong alagang Klebsiella pneumoniae sa laboratory. So yan. Ito na lang yung natitira nating bacteria sa laboratory. So siya lang yung nakasurvive ng pandemic. So saglit mag-alcohol lang ako. Yan. So for today, we will have the continuation of our lecture, and we will be discussing Proteus. Okay. So Proteus is actually has 4 recognized species. So what can you say about Proteus is that Proteus is non-lactose fermenters. So obviously, if Proteus are non-lactose fermenters, nung pinakita ko sa inyo kanina is an example of lactose fermenters. So sila yung magproduce, syempre, when you say lactose fermenters, sila yung magproduce ng pink colonies. But since Proteus are non-lactose fermenters, you would expect them to have colorless colonies or white colonies in McConkey agar. Okay? So but they can ferment glucose, sucrose, and galactose. Okay? So they possess these antigens, OX2, OX19, and OXK, wherein the letter K here stands for the Kingsbury strain. Okay? So why are they important? Proteus antigen is actually being used for Will-Felix test. And Will-Felix test is a serological test for typhus fever. And typhus fever is caused by rickettsia. So I have here some tubes. Okay, so this is the Proteus OX19 antigen. And then I have another here. I have another one here. This is the Proteus OX2 antigen. And I also have here the Proteus OXK antigen. So these particular antigens are actually being used for Will-Felix test. So ang gagawin natin is that we'll extract blood from the patient, and then we centrifuge the blood and get the serum. And then we add 1 drop of this antigen to the serum. And if there's agglutination, it means that that patient is positive for typhus fever. Although Will-Felix test is no longer popular, and some laboratory may even consider this as obsolete already. So you might be wondering, Proteus and Rickettsia are different bacteria. Yes, you're correct, no? So Proteus and Rickettsia are different bacteria. So, however, they found out that those patients with typhus fever will actually have they were able to isolate Proteus bacteria from the urine of patient with typhus fever. So vaguely somehow they utilize the Proteus antigen to determine if the person has antibodies against rickettsia. So that's the reason why it's really not specific. But nonetheless, it's nice to know that Proteus antigens, Proteus antigens can be utilized for Will-Felix tests. Okay. Now, one of the most important characteristic of Proteus is swarming. So this particular thing that you see here is an example of swarming phenomenon. So when you say swarming phenomenon, as you can see here, you originally planted the bacteria here, but the bacteria can actually swarm. Okay, and this somehow indicates that they are motile, but not all motile bacteria can actually exhibit the swarming phenomenon. So on blood agar plate, it says here, this looks like someone pressed an orange skin on the plate. And on EMB, iosine methylene blue inhibits swarming, and it looks like ripples when a rock is thrown in water. Diba pag nagbato kayo ng rocks sa water, so nagkakaroon siya ng ripple effect. So ganyan yung itsura daw ng swarming. Okay, now one of the most important characteristics of Proteus is its ability to produce urease. Okay, urease is an enzyme which can hydrolyze urea. And one of the product of that hydrolysis is ammonia. And this ammonia will make the urine alkaline. Okay? So if the urine has an alkaline pH on a distinct test, then Proteus can be, of course, be considered as an etiologic agent of that particular urinary tract infection. Okay? So we consider Proteus as an opportunistic pathogen. Okay? So we consider Proteus as an opportunistic pathogen. So here again is the appearance of the swarming phenomenon. So you can actually see that the organism swarms once you have planted it here, so they can actually swarm. Okay. So the isolated organisms, so there are two important species, okay? So we have the Proteus vulgaris and the other one is Proteus mirabilis. So they are isolated from urine, wound, tear, and bacteremic infection. Now, one of the most important characteristics of Proteus is the Dines phenomenon. So when we say Dines phenomenon, there is actually an area of demarcation between two species of Proteus. So for example, if this is Proteus vulgaris, and then this is Proteus mirabilis. So you would notice that there is actually an area of demarcation. So this particular area of demarcation between different species of Proteus, this is what you call the Dines phenomenon. So when you open the incubator and there's a Proteus in the plate, you would notice, you'd really smell a burnt chocolate or gunpowder smell. So amoy pulbura. And Proteus can produce hydrogen sulfide. So as we all know, if hydrogen sulfide is present, then there will be what? Blackening at the PSI tube if hydrogen sulfide is present. And if urease is present, the culture medium where we do urease test is called Christensen urea medium. And a urease positive organism will produce a deep pink color. So if the organism is urease positive, it will produce a deep pink color. Okay, so that is for Proteus vulgaris and Proteus mirabilis. Okay, so how do we differentiate Proteus vulgaris from Proteus mirabilis? So Proteus vulgaris is positive for indole, but negative in ornithine decarboxylase. But Proteus mirabilis would have an opposite result. So it is negative in indole, but positive for ornithine decarboxylase. So that's how we differentiate these two important species of Proteus. Okay, aside from vulgari and mirabilis, we also have Proteus veneri, which can also swarm in selected medium, and Proteus mixophaciens, which could be isolated from gypsy moth, and they would produce the so-called large slime colonies. Okay, so aside from Proteus, we also have the Morganella species. Okay, so one of the most important characteristics or important organisms for Morganella is Morganella morgani, which can cause urinary tract infection and also a possible agent of diarrhea. And it is almost, the characteristic is almost similar to Proteus. So as you can see, the PSI is K over A with H2S. So you can actually see hydrogen sulfide because of the blackening. This is the Christensen urea medium. So Christensen urea medium. So if it's urease positive, it will produce a deep pink color. And this is citrate utilization test. So this is negative because green is negative for citrate and Prussian blue is positive for citrate. SIM stands for um, sulfide indole motility. So it's positive for indole because of the so-called red ethereal layer. Okay, so that is Morganella morgani. Okay, so Providencia. So under the Providencia species, we have Providencia retgeri, which is a urinary tract pathogen. Also, they are occasionally implicated in nosocomial outbreaks or whatever outbreaks that involve hospital infections. Okay, so that is for Providencia retgeri. And then for Providencia stewarti, it is implicated in nosocomial burn unit outbreaks, also isolated in urine cultures, though Providencia cannot exhibit swarming, but they are significant because of the fact that they are highly resistant to antimicrobial. So you do not want to get infected with Providencia stewarti. So aside from them, we also have alkalifasciens and rustigiani. So these are the different tests that would allow us to differentiate Proteus, Providencia, and Morganella. So noteworthy to mention are the indole test. So yung veneri and mirabilis are negative, while the rest are positive. All of them are positive in methyl red, but almost all of them are negative in Wolfsburg scours. So tingnan natin yung Christensen urea. So lahat sila positive except for the Providencia. So yung Providencia, negative for urea. But Morganella is positive. So that's why it says here, Morganella resembles Proteus. Okay, who are capable of swarming? Only Proteus are capable of swarming, but yung Providencia and Morganella, they are not capable of swarming. Okay, so let's talk about Yersinia. So this time, we'll talk about Yersinia pestis. So this is the positive agent of plague. Okay, it is transmitted through the bite of the rodents flee. Not the rodents, but the rodents flees. Okay? However, pneumonic plague is transmitted to respiratory droplets. So pag sinabi natin pneumonic plague, it is human to human. Okay. So they are gram-negative coccobacilli resembling a safety pin appearance and bipolar in staining. So CIN agar will help isolate Yersinia species. Yung pinaka-significant dito that almost wiped the population of Europe in the 14th century was the Yersinia pestis. So Yersinia pestis is negative for motility above 22 degrees centigrade, negative for ornithine decarboxylase. But they are negative for Yersinia enterocolytica is positive for motility at 25 degrees centigrade at room temperature, and they are also positive for ornithine decarboxylase. Yersinia pseudotuberculosis is positive for motility at 25 degrees centigrade, but negative for ornithine decarboxylase. So ngayon, medyo syempre, bibihira na natin makita yung Yersinia pestis, and sana wag na, because this particular bacterial infection at that time almost wiped out the population of Europe. So again, these are the biochemical tests that will allow us to differentiate different species of Yersinia. Okay, and let's talk about the Yersinia pestis. There was this 5-year period of pestilence during the Middle Ages. Black Death. Actually, there were 3 notable pandemics of Black Death or Yersinia. Okay, it first appeared in 16th century, and then it reappeared in 14th century, and also reappeared in 17th century. So ito yung mga pandemic caused by Black Death or Yersinia pestis. At that time, parang maraming namatay, primarily because antibiotics haven't been invented during the time of that pandemic. And they didn't know what causes the infection. No? So there are 2 types of plagues. The urban plagues, which had actually occurred in Middle Ages, and it is actually transmitted to rat population known as the fleas. And then the sylvatic plague, and sylvatic plague is actually due to mammalian reservoir. Okay? So yung mga mammalian reservoir na to are found in forest. Okay? Kaya hindi siya urban. It's sylvatic plague. Okay? So the endemic reservoir are fleas. Rats. So yung rats, meron silang fleas. So the fleas are the one that carries the Yersinia pestis. So eventually, fleas actually came from these other hosts. So pwedeng squirrel, chipmunks, basta mammalian reservoir. So during the time of pestilence in Europe, namatay na yung mga mouse. So namatay yung mga mouse. So the fleas actually has beaten the humans. So kapag nakagat yung humans ng fleas na mayroong Yersinia pestis, this is what you call bubonic plague. So when you say buboes is actually a painful swelling of the lymph nodes. So magkakaroon ngayon ng painful swelling of the lymph nodes at the groin and then at the axilla. Sa kilikili. Tapos, sa lymphatic system muna, until such time na magkakaroon na ng sepsis yung pasyente, and then mapupunta sa lungs. So pag nagkaroon ng napunta sa lungs, so magkakaroon na ngayon ng respiratory droplets. So yung respiratory droplets, possible na yung human to human. Kapag human to human infection, ito na yung tinatawag natin na pneumonic plague. Kasi yung lungs na yung affected dito. Okay? And mataas ang mortality rate ng pneumonic plague. Okay? 90% yung mortality rate niya. So, wala talagang nakakaligtas dito sa pneumonic plague because at that time, antibiotics have not yet been invented. Okay? So, again, urban plague is transmitted by fleas from rats. Sylvatic plague has a reservoir of wild animals such as rabbits, rats, prairie dogs, and a variety of flea vectors. So, ganito yung PPE nila nung during the 18th century na nagkaroon ng plague in France, in Marseille, in France. So, makikita ninyo rito na talagang since wala pa yung PPE natin ngayon, so para silang mga ibon kasi ito yung beak kasi ayaw nilang maamoy kasi akala talaga nila yung plague is transmitted through miasma or blood or pag sinabing miasma kasi noong unang panahon wala pa kasi silang knowledge in bacteriology. Akala nila bad air yung nagtatransmit ng plague. So tinatakpan nila yon. And since maraming decaying bodies, meron silang mga herbs dito sa dulo para hindi nila naamoy. So, nakakatulong din yon kasi nga hindi nila na-inhale yung bacteria. Tapos, eto, bakit sila meron ganito? Ginagamit nila yan to touch when the body of a person that they are examining. So, ganito yung itsura ng mga physicians during the time of plague epidemic. So, kahit nga yung nursery rhyme was actually um, came from the plague, yung ring around the rosy. Ayan, parang yung nursery rhyme na yun, ano, um, due to plague, kasi yung rosy refers to the rosy red rashes. Yung posies, ito yung ginagamit nila pagtakip ng kanilang mga bibig. Yung hasha, ito yung cough, kasi nga pag nagka-pneumonic plague na yung pasyente, they would keep on coughing. And then we all fall down, refers to the death. Kasi nga, 90% yung mortality rate ng plague during that time. So, hindi lang pala ngayon nagkaroon ng pandemic. So, even before, nagkaroon na ng pandemic, no? So, there are two forms of plagues. So, we have the bubonic plague. The incubation is about 7 days from the bite of infected fleas, no? So characterized by painful buboes in groin or in axilla. And then, pneumonic plague has a shorter incubation period and fever, malaise, these are the pulmonary signs, and then 90% yung mortality rate. So yun. So later on, we will watch a documentary on plague. So sana mag-work yung mamaya. Okay, so but before we watch the documentary, so let us actually discuss first other species of Yersinia. So we have Yersinia enterocolitica. Yersinia enterocolitica, the reservoir are swine, cats, and dogs, and it is characterized by acute gastroenteritis simulating appendicitis. Akala ng tao meron appendicitis. So, and the reason why you got this infection is the ingestion of fecal material, particularly from this animal reservoir. So it will primarily affect children between 1 to 5 years of age, immunocompromised adults. Okay? So Yersinia enterocolitica is characterized by erythromanodosum. So pag sinabi erythromanodosum, it is characterized by tender red nodules with itching and burning on lower legs or yung shins. And your Yersinia enterocolitis are actually isolated in Scandinavian and European countries because these bacteria are more active in cold climate. So diarrhea may last up to 2 weeks. Okay? And then we also have Yersinia pseudotuberculosis. So yung Yersinia pseudotuberculosis, okay, was actually isolated from rodents, farm animals, and birds. So, kaya sa tinawag na pseudotuberculosis kasi it resembles tuberculosis, but it's not really the actual tuberculosis. So this one is characterized by caseous swellings called pseudotubercles in mesenteric lymph nodes. And the mode of transmission is also attributed to the ingestion of fecal material. So, halos kapareho siya ng Yersinia enterocolitica. So, yun nga, yung caseous swelling is the reason why it's called pseudotuberculosis. So, it is fatal in animals, but human infection is rare. So, fortunately for that. So, however, whenever we are infected, so this one is characterized by septicemia with mesenteric lymphadenitis. Okay, so moving on, we have another bacteria, and this time we are discussing Edwardsiella. So under the Edwardsiella, we have the Edwardsiella tarda, which is the only known pathogen. So Edwardsiella tarda is characterized by urea negative, lysine decarboxylase positive, but these organisms are positive in hydrogen sulfide production and indole production, but they cannot actually utilize citrate. So Edwardsiella is actually implicated in bacteremia and in wound infections. So there are three important species of Edwardsiella. So these are Edwardsiella tarda, which can cause opportunistic wound infection and bacteremia, the one that you can see here. And then Edwardsiella hoshinae is isolated from snake, birds, and water, and in water, and Edwardsiella ictaluri, which can isolate them in fish. So as you can see, they can cause infection in these particular kinds of animals. Okay. And then Erwinia and Pectobacterium, these are considered as plant pathogens. Okay. So let's talk about Citrobacter species. So under the Citrobacter species, we have the Citrobacter frundi. Okay, so this one is the most common, and it can actually cause nosocomial UTI. So a UTI that is attributed to hospital confinement or being exposed in the hospital. So aside from that, Citrobacter frundi may also cause pneumonias, endocarditis, and in rare cases, intra-abdominal abscesses. So parang yung mesentery natin can also be infected with this kind of bacteria. So aside from Citrobacter frundi, we also have Citrobacter cosseri, formerly known as Citrobacter diversus. So now, ang tawag na sa kanya is Citrobacter cosseri. So this one is implicated in nursery outbreaks of neonatal meningitis and even in brain abscesses. So parang nagkaroon ng pus formation sa loob ng brain. Okay? So, how do we identify Citrobacter? Citrobacter is sometimes mistaken for Salmonella. Okay? But they have this weakly urease activity and they can ferment lactose. Unlike Salmonella, which cannot ferment lactose. Citrobacter can also grow on Simon citrate and they are methyl red positive. So similar to Salmonella, but not exactly the same. Primarily because Citrobacter frundi can hydrolyze urea, but they cannot decarboxylate lysine. Unlike Salmonella, most Salmonella cannot hydrolyze urea, and most Salmonella can decarboxylate lysine. So these two are the distinguishing characteristics of Citrobacter and Salmonella, so that we will not be mistaking them from one another. Okay, so these are some of the important species of Citrobacter. So we have Citrobacter frundi implicated in nosocomial infection, UTI, pneumonia, and intra-abdominal abscess. Citrobacter diversus, now known as Citrobacter cosseri, implicated in nursery outbreaks, neonatal meningitis, and brain abscess. And Citrobacter malonacticus, frequently found in feces, blood, and in wound infection. Another species of bacteria that is similar to Salmonella is Arizona hinshawi. it is similar to Salmonella and may cause gastroenteritis or sepsis, but unlike Salmonella, Arizona is considered to be as a slow lactose fermenter. So, aside from the previously mentioned bacteria, we have other or new genera, no, of Enterobacteriaceae. So we have the Providencia, Vitusilla, Sedicea, Morgonella, Pluiveria, Leclercia, and Leminorella. Okay, so these are the new genera. Molerella, Obesum bacterium, Photorhabdus, Pragia, Ranella, Patumella, Trabulciella, Cenorhabdus, and Enella. So, how do we determine, how do we diagnose enterics? Of course, very important that we do specimen collection and transport. So isolation and identification would really play an important role, such as pag sinabi natin isolation, this would involve direct microscopic examination. So guys, when you say direct microscopic examination, it means that we directly examine the specimen. So how do we examine it? Usually by means of gram stain and other special um, microscopic techniques. Yan. Pero pag sinabing natin culture, we usually use selective and differential media. Okay, so like what we have discussed from the previous lecture, we are using mostly McConkey agar. Pero aside from McConkey agar, we can also use Hectorine enteric agar, XLB, so the one that I've mentioned to you before. And of course, identification. So nowadays, pwede na kayong gumamit ng mga PCR, MALDI-TOF for identifying this particular enterics. And then we can also screen stool culture for pathogens. Okay, so medyo mahirap yung screening for stool culture. So usually nilalagay muna natin sila sa enrichment broth. Ano ba yung mga example ng enrichment broth? Selenite F for Salmonella. And then alkaline peptone water for cholera. So these are examples of enrichment broth. And then of course, serotyping. So we can do the serologic grouping or by means of serotyping. Okay, so with that, we can actually have the algorithm for the isolation and identification of the enterics. So yung first is yung TSI, yung triple sugar iron, and I already taught you how to identify them. So through the TSI, we can have 5 major groupings. So we have K over A with H2S. So ibig sabihin nito, red over yellow with blackening. Kasi pag tandaan niyo guys, kapag positive sa H2S or hydrogen sulfide, ibig sabihin merong blackening. Pag sinabi natin A over A with H2S, yellow over yellow with blackening. Pag sinabi natin K over A with H2S, ang ibig sabihin niyan is red over yellow but no blackening kasi nga H2S negative. Pag sinabi naman natin A over A with H2S, ang ibig sabihin naman nito is yellow over yellow, no blackening. Kapag K over K naman, it is when no fermentation has taken place. So, red over red. So, mga yan. So, ngayon, kailangan sa non-lactose fermenters na tayo titingin. Okay, ito naman, or sa mga non-fermentative bacteria tayo titingin. Kapag K over K, which is another topic na to. Okay, so guys, for Proteus mirabilis, K over A with H2S, so we have Proteus mirabilis, Salmonella, Citrobacter, and Escherichia tarda. So we can do the PAD test. Pag sinabi natin PAD test, phenylalanine deaminase. So it is the ability of deamination of phenylalanine. Pag sinabing deamination, removal of amino acid. And what amino acid is being removed? Phenylalanine. Kaya siya tinawag na phenylalanine deamination test or PAD test. So, ibig sabihin, yung mga bacteria katulad ng Proteus mirabilis is PAD positive. So, ibig sabihin, kaya niyang alisin yung phenylalanine amino acid. Pero yung Salmonella, Citrobacter, Fundi, and Escherichia tarda, they're PAD negative. So, we do further test yung lysine decarboxylase. Ang ibig sabihin ng decarboxylase, removal of carboxyl group. Ito yung COOH. So, yung Salmonella and species positive sila. So, ibig sabihin, kaya niya mag-remove ng carboxyl group. Okay? Yung Fundi and Salmonella, negative. Yan. Tapos ngayon, to differentiate Salmonella from Edwardsiella, we do the indole test. So, Edwardsiella tarda is positive, Salmonella is negative. Ayan. So, ito naman, sa second group, we do the PAD test and only Proteus vulgaris is positive here. So, the rest are negative. Kapag K over A with H2S, ang dami pa lang choices. So, again, we do the PAD test. So, ito yung mga positive. Providencia redgeri, Morganella morgani, Providencia stuarti, and Providencia alcalifaciens. Tapos, to further differentiate them, we do citrate test. So, yung Providencia is positive, Morganella is negative. And then, yung mga negative naman dito, we do the motility test. So, E. coli, Citrobacter, and Serratia are positive for motility. Shigella and Yersinia, they are negative. Kapag A over A with H2S naman, ito yung group na to, ito yung mga possible choices. So, we do the INVIC test. So, ano nga yung INVIC? I stands for indole, M stands for methyl red, and then V stands for Wodge-Proskauer, and C stands for citrate. Okay, so apat na biochemical test kapag sinabi natin INVIC. And E. coli has a result of positive, positive, negative, negative. So ito yung result ng E. coli. Pero kapag ang result naman ay baligtad, negative, negative, positive, positive, so we do the motility. So Enterobacter and Serratia is positive, Klebsiella is negative. Tapos, para ma-differentiate yung Enterobacter from Serratia, we do the DNase test. Serratia is positive for DNase, Enterobacter is negative for DNase. So, mahalaga talaga yung table na to. Swerte nga ninyo, wala kayong hindi maswerte. But one of the things na ginagawa namin sa lab is binibigyan namin sila ng unknown bacteria. And yung mga estudyante, yung mag-identify ng mga bacteria na yun. So, sayang, hindi natin nagawa. But nonetheless, prayerfully magawa natin yun pagdating ng campus-based internship. Okay?